Hello. Today we're going to be looking at the paper chromatography of soy sauce and amino acid. Now, to do this we're going to do two separate chromatograms. We're going to do first of all a chromatogram of two different types of soy sauce. We have light and dark soy sauce, which contains amino acids. We're then going to do a comparison chromatogram of 3 amino acids which we know to be present in soy sauce, and we'll then compare the two afterwards. So first of all we'll go through how we're going to set up our chromatograms. So once again we're using chromatography paper. It's important that we're very careful how we handle this and that we don't put our fingerprints and dirt and things all over the chromatogram. We only handle it by the edges. The reason for this is the grease and dirt can affect how substances flow up and down the paper. So the first thing we're going to do is we're going to, from the bottom, mark a light line in pencil. Now it has to be in pencil and it must be a light line, 10 mm from the bottom. Okay? You're not pressing on particularly hard, you're just sketching a very light line while trying not to get your fingers all over the paper. Okay, now we're doing it 10 mm up, so that's where we're going to put our sample. Okay, and that way we can also make sure that they're all starting from the same point and that point is above the level of our solvent. Now on one chromatogram we're going to run 2 separate samples. So what we're going to do is we're going to divide it into 3. So in this case it's 8 wide, so we can do about 2.5 and at 5 we're just going to put a little mark. In this case it's light and dark soy sauce, so I'm going to label one as an L and the other as a D. Again, gently and in pencil. The other one we're going to use for amino acids. Now we have 3 separate amino acids, so I'm going to divide this one up. This one's slightly narrower, so I'm going to do 2, 4 and 6. So we're just making sure that they're evenly spaced and they're far enough apart that as they run they're not going to interfere with each other. So I have aspartic acid, which I'm going to mark with an A, glutamic acid, which I'm going to mark with a G, and leucine, which I'm going to mark with an L. Okay, so when we've done that we're going to prepare our soy sauce samples. Now soy sauce is quite a dark, thick liquid to start with. So we're going to do a dilution of this, okay, with distilled water to 1 in 10, which means that we're going to add 2 ml of the soy sauce to 18 ml of water, okay, so that we can dilute it so that we're not using too concentrated a solution that it's going to get stuck in our capillary tube, because it is quite, particularly the dark soy sauce, it is quite thick and gloopy. So we've got our light, okay, and we're going to do our dark. Again, it's exactly the same thing. It's 2 ml to 18 water. Okay, I'm using a different measuring cylinder this time, but this one is really a lot thicker and gloopier and stands a good chance of getting stuck in our capillary tube. Right, so we're just going to make sure that it's evenly mixed, so that it's all nice and mixed with the water. Okay, we're going to take our chromatogram for the light and dark. Sticky, horrible stuff. Right, we're going to take our chromatogram for the light and dark. We're going to use a capillary tube for each of the 2 samples. Okay, one for light and one for dark. And we're going to put the capillary tube into the solution put our finger over the top of the capillary tube so we've got some liquid in it, and then we're just going to put a small spot, a couple of mil diameter, of the soy sauce where it says L on our line, and we're going to do the same for the dark where it says D. Now you don't want a huge, it will spread out, okay? You don't want to try and get it to spread out too far, because obviously it will then start to run and interfere with each other. That's one of the reasons why we're splitting it across 2 chromatograms. So, pre-prepared earlier. And so now we're going to have to spot these more than once. So while that's drying, I'm going to prepare the next one. So A, G and L, we've got our 3 amino acids. Amino acids are not particularly soluble. Okay, so there is going to be some powder and residue left in the sample. Okay, so we have to be careful that we're picking up the liquid bit, otherwise we'll end up with crystals sat on the top of our surface of our chromatogram, or we'll end up blocking up our capillary tube. And we're going to have to do the same. We're just going to spot on exactly the same way. We're just going to spot on in exactly the same way. Aspartame onto the A. Glutamic acid onto the G. And leucine onto the L. Now each one of these will need to be spotted and allowed to dry at least 5 to 10 times. So while that one's drying, I can then go back to doing my light and dark soy sauce. Exactly the same procedure again, and we just repeat the spotting. Okay, and hopefully by the time we've done that, the other one will be dry and we can just keep swapping them over. Now it's important that you do this because you've got to build up the concentration of the chemicals on the paper. If there's not enough chemical, it will not develop when we have ran the chromatogram. Okay, so and the more on there, the more chance you have of being able to see the bands. Okay, and we just, as I say, just keep doing it until you've got a reasonable build-up. Now, while that's happening, we can also start preparing the next stage. So we're going to run our chromatograms into 2 beakers. We're going to use a particular solvent. This is a butanol ethanoic acid water solvent with 4 parts butanol to 1 part ethanoic acid to 2 parts water. Okay, it will be flammable and corrosive, so we need to be careful and make sure we're wearing our safety glasses when we're using this. Okay, now again, we're only putting a small amount in the bottom of our beaker to about 5 ml depth, because we need to make sure that the solvent level is below that of our line and where our sample is. Okay, we're going to put lids on them because obviously they're solvents and we don't want the fumes everywhere. So it's important that we keep that solvent level below our line. If the solvent level comes up to our line, the material that we've deposited on the line, all of our sample, instead of flowing up the paper, will flow out into the solution. And that is the last thing we want, because then that means we have to start all over again. I'm just trying to quickly build up sufficient on each one of these so that we can run it. Okay, now when I've got the samples done, I'm going to place them with my fingers, put my fingers down into the solution, making sure that the level of the solvent does not come up to the line. Okay, and we're going to continue this video in part 2 when we're going to look at the results and how we develop them. Thank you.